Hey guys, welcome back. Today I'm going to be covering this MCAT passage called What Causes Red Blood Cells to Sickle? So the passage goes, sickle cell disease is an autosomal recessive disease commonly found in African populations. The disease gets its name from the fact that patients' red blood cells become sickle-shaped when passing through the capillaries of the metabolically active tissues. These red blood cells become frail and can rupture long before their normal lifespan. The sickled red blood cells block capillaries and inhibit red blood cell function, causing severe anemia in sufferers. Okay, so this was all preliminary information. There was nothing really useful here. Okay, the next paragraph says, the most common hemoglobin in adults is hemoglobin A, labeled HbA. However, patients suffering from sickle cell disease are homozygous for the allele coding for the abnormal variant of hemoglobin, which is HbS. So one important piece of information here was that the fact that sufferers were homozygous for the hemoglobin allele. And due to this missense mutation, the gene encoding the beta subunit of hemoglobin. Okay, so we also now know that it's due to a missense mutation in the gene that encodes the beta subunit for hemoglobin. This missense mutation replaces glutamic acid with valine at the 6th position of the beta globin chain. Okay, since this is a chemistry passage, this is where you should be focused on. So we had a glutamic acid residue that was changed into a valine residue. So let's keep that in mind. It says the absence of a polar amino acid at this position promotes a non-covalent aggregation of hemoglobin in a low oxygen environment, which distorts the red blood cells into a sickle shape and decreases their elasticity. Biochemically, the low oxygen environment causes the beta chain of the neighboring hemoglobin molecules to hook together. So the low oxygen environment causes the beta chain of neighboring hemoglobin to hook together, becoming rigid and polymerized. These cells fail to return to their normal shape when oxygen is restored, and thus fail to deform as they pass through narrow blood vessels, leading to blockage in capillaries. Okay? So there was a lot of useful information in this passage, mainly what I've kind of underlined here. So let's continue. In vitro studies of deoxygenation and reoxygenation of sickle cell hemoglobin indicates that the process is reversible. The hemoglobin molecule polymerizes and form crystal as oxygen concentration is lowered, but as oxygen concentration is increased again, the hemoglobin molecule can depolymerize and return to their soluble state. Okay? So this seems to be an important diagram. So we can convert between the non-sickled form of hemoglobin to the sickled form of hemoglobin, depending on how much oxygen is available. Okay? It says the discovery of sickle cell mutation came from an isoelectric focusing experiment, a type of electrophoresis that separates complex mixtures of large molecules by applying an electric current. Hemoglobin taken from patients suffering from sickle cell anemia, individuals who were heterozygous for the abnormal variant, and those who did not have sickle cell allele were subjected to isoelectric focusing. The following results were obtained. So they did an isoelectric focusing experiment, and this is what they found. The normal hemoglobin allele is over here. The sickle cell allele is here. Now, we know from our second paragraph that when you have sickle cell anemia, essentially a glutamic acid is swapped with a valine. So this is a polar residue because it has the glutamic acid, and this is non-polar because it's been swapped with the valine. And when you're heterozygous, you have kind of peaks at both. So you have both polar and non-polar residues present, similarly with a 50-50 mixture. So now let's get to the questions. It says, which of the following amino acids, if present at the 6th position of the beta globin chain, would yield a non-covalent aggregation of hemoglobin in a low oxygen environment? Well, again, based on the second paragraph here, it says, it says glutamic acid was replaced with a valine, and this is what essentially called the aggregation of hemoglobin residues. So we have to find another amino acid residue that is similar to valine, such that it is non-polar, because they want a non-covalent aggregation. Well, we know threonine and serine both have OH groups. So it can't be either one of them. And we also know that aspartic acid is an acid, meaning that it is has an acidic residue, which means it is also polar. So we have these three residues that are polar. Now isoleucine, when compared to the other three, is the only non-polar residue within this list. So it's most likely to be the answer. So let's check, and it is. Next question. How can the amount of soluble non-sickled hemoglobin be increased in an in vitro study? So the options are reduce the oxygen concentration level, reduce the amount of sickle cell hemoglobin polymers, reduce the amount of soluble non-sickled hemoglobin, or introduce additional sickle cell hemoglobin polymers. So I think we should focus on this whole diagram to answer this question. Now it says we want to increase the amount of non-sickled hemoglobin. So we want to create more of this. Well, how can we create more of this? We know based on Le Chatelier's principle that we could either decrease the amount of non-sickle celled hemoglobin, or we could increase the amount of sickle celled hemoglobin. If we decrease non-sickle cell hemoglobin, the reaction shifts to the left, or if we increase the amount of sickle cell hemoglobin, the reaction again shifts to the left. Now, with regards to the oxygen concentration present, one of the options stated that reduce the oxygen concentration level. Well, if we were to reduce the oxygen concentration level, the reaction would actually shift to the right, because low oxygen is the conditions for sickle cell hemoglobin polymers to form. Which means we know that this isn't the answer. What about reducing the amount of sickle cell hemoglobin polymers? Well, if we reduce the amount of sickle cell hemoglobin polymers, it's going to push the reaction to the right, again, due to Le Chatelier's principle. So we know that's incorrect. What if we reduce the amount of soluble non-sickle cell hemoglobin? So yes, we could reduce the amount of non-sickle cell hemoglobin, so let's keep that for now. What about introducing additional sickle cell hemoglobin polymers? So yes, we could also increase the amount of sickle cell hemoglobin. Now, between these two options, if we want to have more non-sickled hemoglobin, should we be decreasing the non-sickle cell hemoglobin, or should we just be increasing the sickle cell hemoglobin? And logic should dictate that we should just add additional sickle cell hemoglobin polymers. Because if we were to take away non-sickled, we would have to initially decrease the concentration of non-sickled for it to subsequently increase by a bit. So that's kind of counterintuitive, so we should avoid that. So I'm going to say the answer is to introduce additional sickle cell hemoglobin. This question asks, how does the beta chain of the neighboring HBS molecule cause hemoglobin to aggregate? So remember, HBS is the aberrant form of hemoglobin when the glutamic acid has been replaced with a valine. Well, if a glutamic acid has been replaced with a valine, and we know that valine is nonpolar. Well, things that are nonpolar typically undergo a very specific type of aggregation that you guys should know for your MCAT, which is hydrophobic aggregation. So the option choices are the interaction between glutamic acid and other hydrophilic amino acids create a hydrophilic pocket leading to hemoglobin aggregation. Well, we know this can't be true because HBS molecules don't even have a glutamic acid. As the question stated, that HBS variants have the glutamic acid replaced with a valine. So we know this isn't right. Option B states, the interaction between valine and other hydrophobic amino acids create a hydrophobic pocket leading to hemoglobin aggregation. So this sounds accurate. Glutamic acid hydrogen bonds to other amino acids. Well, again, we know just based on the fact that this option states glutamic acid that it's incorrect. And the last option is valine causes hemoglobin molecules to exhibit increased van der Waals forces due to hemoglobin's increased size. Well, here's the thing. Replacing one glutamic acid with one valine doesn't necessarily increase the size of the hemoglobin itself. Rather, it just changes its chemistry. So this answer isn't right either, because hemoglobin, even in the HBS variant, would be roughly the same size. So we can safely assume that this is the answer. What does the distribution in the isoelectric focusing experiment indicate about heterozygous individuals? Well, if we look here, as I mentioned when I was explaining the passage, heterozygous individuals seem like they're experiencing a peak at both the normal and sickle cell variant, meaning they exhibit parts of both with regards to their polarity and their isoelectric distribution. So the options are they exhibit two different types of hemoglobin proteins separated from each other in the electrophoresis by a difference in length. Well, maybe. Let's keep that for now. They exhibit multiple different types of hemoglobin proteins. Well, again, based on this graph, we only see two peaks. It's not like we're seeing a bunch of peaks. If it had multiple peaks, it would kind of look like that. But instead, we only have two distinct peaks. So we know that's incorrect. They exhibit multiple different types of hemoglobin proteins. Well, again, we don't have multiple different types of we simply have two different types, 1 and 2. And they exhibit two different types of hemoglobin proteins separated from each other in electrophoresis by the difference in their charge. Well, yeah, because we know that in hemoglobin, again, glutamic acid is replaced with a valine. So something that is charged is replaced with something that is nonpolar and uncharged, meaning this is charged and this is non-charged. And in the heterozygous individual, we see aspects of both. So this separation in isoelectric focusing specifically is due to charge, not due to differences in length. There's different types of experiments and different types of electrophoresis that separate based on length, and isoelectric focusing is not one of them. And the last question states, how would the distribution of hemoglobin proteins in the 50-50 mixture be different if a higher voltage difference were applied between the two electrodes? Well, again, we know based on this graph that they're separating based on their charge. So we have highly charged and we have lowly charged. If the potential difference or the voltage difference between the two ends was bigger, we can simply expect there to be a larger gap between the two places as they will be separated more. So your option choices are the bimodal distribution would become more pronounced as the electric field would be more effective in imparting a charge to the protein. Well, maybe. That sounds somewhat right. The bimodal distribution would become less pronounced. Well, like I mentioned here, since there's a higher voltage being applied, we would expect the two poles to kind of separate further and to become more pronounced. So the fact that this is saying less pronounced leads me to believe that it's incorrect. Option C states the bimodal distribution would become more pronounced as the electric field would become more effective in separating hemoglobin proteins from one another. Again, maybe. And the last option says the bimodal distribution would become less pronounced as the electric field would become less effective in imparting charges to proteins. Well, again, it would be more pronounced, not less. So between these two options, I think in order to narrow it down to one final option, you have to understand what isoelectric focusing is. Now, in isoelectric focusing, it's not as if the actual experiment involves imparting a charge to the protein. Rather, it separates based on the charge that the proteins already have. So this would be highly charged and this would be lowly charged. So it's not really a matter of imparting the charge to the protein, rather than how different the charges between the separate proteins are. So this is the more accurate answer, where the difference would become more pronounced as the electric field would be more effective in separating hemoglobin proteins from one another. And that is right. Well, I hope this has been helpful. I will try posting more of these MCAT videos if people do find it helpful. And if you have any other suggestions, please leave it in the comments below. Thank you.